Hello everyone, and welcome to episode 110. This is the second episode in our series on the arachnids, and today we're going to explore a few more of the relatively basal lineages in this unique arthropod clade. Now, if you haven't listened to the previous episode 109 on arachnid evolution, I strongly recommend that you do that before listening to this one. Episode 109 was not just an exploration of arthropod and early arachnid evolution. It also looked at living representatives of some of the most primitive basal clades of arthropod, like horseshoe crabs and tardigrades, and basal arachnids like Pycnogonida sea spiders and Apillionese harvestmen. I also clarified the issues with the current understanding of arachnid phylogeny. To summarize, genetic analyses over the last 20 years or so have given inconsistent results, with the placement of and relationship between certain groups changing depending on what sections of the genome are sequenced and compared. A clear, concise, consolidated tree of life for the arachnid monophyly hasn't quite been developed yet, but progress has been made, particularly in the last decade. Currently, one of the biggest issues is the organization of several basal clades in the arachnid class. The precise order of their evolutionary emergence and their precise relation to each other hasn't been conclusively determined. I discussed 3 of these lineages in the last episode: the Apillionese harvestmen, the Ricynulae pseudospiders, and the Solifugi camel spiders. Take note that despite these organisms having spider in their common name, or there's a widespread perception that they're spiders, like the harvestmen or daddy longlegs, none of these arachnid orders are actually true spiders. For now, we're going to look at some of the other orders branching out at unclear points from this messy, cryptic, primitive era of early arachnida. Specifically, we're going to look at the Acariformes and the Parasitiformes, 2 distinct superorders that are nonetheless grouped together as mites. This is a pretty good example of a paraphyletic clade, created by changing standards in technology and understanding of animal phylogenies. Miniclades, from crustaceans to reptiles to flowering plants and fungi, were at first organized taxonomically according to their morphological features, to their visually apparent similarities both inside and out. As this kind of macro-scale visual comparison could be performed without a microscope or genetic sequencing, these morphology-based phylogenies were basically like the first edition or the first version, the first attempt to develop some kind of ordered taxonomy of the animal kingdom. Then, with time, over the centuries, we built microscopes, and we could look at cells and microscopic features of the anatomy. And then we developed genetic sequencing and could look at the molecular code in the genome itself. This molecular analysis, this genomic data, provided a much more robust, reliable, and accurate way to create phylogenies. And as we sequenced the genomes of various organisms, we came to realize that while phylogenies based on morphology were more or less accurate for some groups, for many other groups, like crustaceans and mollusks and insects, they were way more inaccurate than we thought. Species, genera, families, orders, even entire classes were moved around, entire phyla redefined. In this way, a large group of morphologically and often behaviorally similar animals may be grouped together, only for subsequent genetic analysis to demonstrate clear distinctions and low relatedness between some of the subgroups within the large group. This is what happened to the Acariformes and the Parasitiformes, which were once all described as mites of the subclass Acari. It was only after molecular and genetic analysis that scientists understood that this Acari subclass was, in fact, 2 distinct groups that were somewhat closely related to each other, but not closely enough to justify grouping them together as one monophyletic group. They're not sister lineages. So the Acari subclass became obsolete, and the 2 genetically distinct groupings were then recognized for what they were: individual arachnid superorders, Acariformes and Parasitiformes. To give you a brief summary or overview of each clade before we really dive into the meat and potatoes of it all, the Acariformes include over 32,000 described species of mites, which makes it the most species-diverse clade of all arachnids. Acariformes are known as micro-herbivores. Individual grazing mites, because of their small size, will eat very small quantities of plant tissue, like wood or leaves, or fungal tissue like a dense patch of mycelia or a fruiting body. But in large numbers, the collective eating activity of a large number of mites can be devastating for plants. However, they're not exclusively micro-herbivores. Some species are scavengers or detrivores that eat decaying plant and animal biomass, and many species are animal parasites. Now, the Parasitiformes represent another 12,000 species of mites, but as the name suggests, they're much more predisposed to a parasitic lifestyle, to filling parasitic ecological niches. However, just as not all the Acariformes are micro-herbivores, I just mentioned that some are omnivores and some are scavengers and parasites, not all of the Parasitiformes are actually parasites. Some of them are active predators that hunt tiny prey, like even smaller mites that are living in the soil, the leaf litter, and leaf canopies of forested habitats. Other Parasitiformes have a more traditionally micro-herbivorous lifestyle. Instead of parasitizing an animal or hunting one down, they eat plant tissue, like rotting wood and pollen, as well as fungi and animal detritus, like feces and festering corpses. In almost all cases, the mites are small, measured in single-digit millimeters, with bulbous bodies and 8 legs. Their body segments have a broad connection, giving them the appearance of being entirely unsegmented. In many species, they actually are unsegmented. Their body segments have fused together, except for their legs, so they can still bend their legs and move around. But the abdomen, thorax, and head have merged into one continuous, consolidated central body. Now, the mites are actually born with 6 legs, so just 3 pairs of legs, but after several molting events, they'll start to grow a new pair of leg buds, and over time, over several molts, these will unfurl into the 4th pair of legs. During these early stages, as the larva is growing through the juvenile life period, after about 6 molting events, give or take, they'll reach maturity. Many species are also described as having intermediate stages like protonymph, deutonymph, tritonymph, etc. before reaching adulthood, but these are generally names for the discrete stages between each individual molting event, or each instar. The mites have male and female sexes, and they generally engage in sexual replication via spermatophore, although there's a surprising number of asexual, female-only parthenogenic species. In those species that practice sexual replication, the male will typically package his sperm in a wad of mucousy goo, and either the male or the female will then put the spermatophore material into the female's seminal receptacle, which is an orifice that leads to the ovary. Some Acariform groups use direct insemination via a penis instead of a spermatophore. But however it happens, after fertilization and gestation, the female will lay a clutch of eggs, typically on whatever substrate the species lives on. Some bury their eggs in the leaf litter or the moist soil, while others attach them to the underside of leaves or branches and so on. Individual mites eat small amounts of biomatter in whatever habitat they happen to find themselves in. Those that live in the forest tend to graze on the ample vegetation and the fungus in the soil, while they are, in turn, preyed upon by larger, more carnivorously inclined species. Some mites seek out corpses or dung, easy sources of food that don't fight back or run away. Most characteristically, and perhaps most interestingly, many species of mites have evolved to parasitize animals, either by living in their fur or feathers or scales, and eating their oily secretions, or their dead, dried-out flakes of skin or loose scales. We'll get to some of these parasitic and ecological dynamics a little bit later, but first we have to find the path that'll take us there, and to set the stage, we need to look at their evolution. It's clear from the fossil record and genetic analysis that between these two groups, the Acariformes came first. They emerged sometime in the late Silurian or the early Devonian, roughly around 420 million years ago. This is right at the time when the true arachnids began to appear, and shortly before the appearance of the oldest, most primitive Apillionese fossils, suggesting that these Acariformes may represent one of, if not the oldest surviving arachnid lineage. I wouldn't call that a certainty, but it's possible, based on the rather limited paleontological evidence. In a fascinating twist, the Acariform order has many species that are parthenogenic, which means females can auto-fertilize somehow through some mechanism and give birth asexually, in a clade that otherwise reproduces sexually. In some cases, like we've seen with certain lizards, reproducing parthenogenically has led to the emergence of entire populations or entirely new species that are parthenogenic. In the case of these Acariform arachnids, it's suspected that the common ancestor of the clade may have distinguished itself from its peers by reproducing through parthenogenesis. No longer constrained by the need to find a mate, this lineage could reproduce asexually until a large population was generated, in which case they may revert to sexual replication again. There's evidence suggesting that at least some Acariform lineages have adapted parthenogenesis multiple times, possibly during a time of hardship and low population numbers, until enough individuals exist where mutations that enable sexual replication become viable again, only for another time of hardship to push them back into using parthenogenesis. To be sure, this is speculative hypothesizing, based on limited evidence. These hypotheses need to be further tested and explored before any hard claims can be made to this early arachnid, early Acariform evolution. It's just extremely fascinating to think about how this trait may have influenced the emergence and diversification of the Acariformes. Now, with regards to the other superorder, the evolution of the Parasitiformes is much more concise. The oldest fossilized Parasitiformes can be found in amber deposits from the Cretaceous period, dating to barely 100 to 105 million years ago. The actual age of the Parasitiform lineage is certainly far older than that, estimated to be around 300 to 360 million years ago. But even this is significantly younger, at least 50 to 100 million years younger, than the Acariform lineage. And in addition, the fossil Parasitiformes emerged roughly around the same time as many other arachnid lineages, like the Resnulae pseudospiders and the Solifugi camel spiders. The ticks of the Parasitiform order Ixodidae, perhaps the most famous groups of mites due to their blood-sucking parasitism, had a common ancestor that's estimated to have existed around 195 million years ago. The relative youth of the Parasitiform lineage is also supported by the fact that Parasitiform species are much more genetically similar, and they show less genetic divergence amongst each other among various species than the older Acariform lineages do among themselves. There's much more genetic diversity among the Acariforms, primarily because they're an older lineage, and there's just been more time to accumulate mutations and build up that diversity, those divergences. Frustratingly, it's not entirely clear how the Parasitiformes emerged. The data doesn't paint a very consistent picture, and I'm honestly not sure which hypothesis is currently preferred among evolutionary biologists. You'll notice that this is going to be a common theme with the study of mites. For example, there was some work in the 2010s suggesting that Acariformes were closely related to the Solifugi, the camel spiders, and that the Parasitiformes were closely related to the pseudoscorpions. All four clades, it was suggested, share a common ancestor that existed around 425 to 430 million years ago, representing the beginning of this branch of the arachnid group. Taking a step back and looking at the larger evolutionary picture here, this hypothesis suggests that after the emergence of the sea spiders, the arachnid class basically split into two major branches. One branch would produce the Solifugi camel spiders, the pseudoscorpions, and the many thousands of species of Acariform and Parasitiform mites, while the other branch would produce the true scorpions, as well as many pseudospiders and the true spiders of the massive order Arachnid. Now I'm just speculating out loud here, but if you consider that most camel spiders and pseudoscorpions are carnivorous, that suggests that the group's common ancestor was also carnivorous, perhaps some kind of aquatic or semi-aquatic predator that creeped along the edges of verdant, plant-dense floodplains and riparian corridors, where it hunted for tiny bugs and other small critters in the foliage. This hypothetical context presents an obvious pathway by which the lineage could have adapted from their ancestral marine habitat to drier, hotter, more terrestrial habitats, with rivers, these riparian corridors, being temperate, comfortable trails leading uphill, deeper into the harsher, drier landscape. The initially carnivorous ancestor species would adapt to a plant-filled environment, giving rise to many new herbivorous lineages, as various populations adapted to more accessible vegetable food sources. Later, as terrestrial vertebrate animals became more common and diverse throughout all of the dry land biomes, the animals themselves became a new kind of substrate, a new kind of platform that could carry the mites around and provide them living space and food. The emergence of larger, complex animals provided frequent opportunities for the evolution of ectoparasitic lifestyles. I will repeat the fact that the ticks, the most famous blood-sucking parasites on the planet, emerged at least 195 million years ago, which would have put the age of their birth in the early Jurassic period, only after large-bodied animals like dinosaurs and cynodont descendant mammals had re-established themselves after the Triassic-Jurassic mass extinction event. The birds emerged sometime later in the Jurassic, and the mites would quickly adapt to use them as hosts as well. While the fossil evidence is sparse, and the genetic evidence can often be unclear, this very broad strokes explanation for the overall evolution and diversification of mites seems generally plausible, and fits nicely with what little hard material evidence there is. I should clarify that while these ecological hypotheses about the habitats they adapted to and the niches they occupied are robust and well supported, later work on their genetics and evolution has proposed different relationships, with various clades rearranged in subtle but important ways. For example, after that original hypothesis was proposed by a paper in 2014, a paper that came out about 5 years later found that the pseudoscorpions were actually more closely related to both the Acariformes and Parasitiformes than they actually were to the true scorpions. And those other basal groups, like the Solifugi sun spiders and Ricinulae pseudospiders, well, they were put in a whole different branch of their own. These groups have been historically problematic for researchers. Apparently, the Solifugi and the Ricinulae keep getting shuffled around depending on the genes in the dataset being used. For example, their relationship to the Opiliones, the harvestman spiders or the daddy longlegs, it seems to fluctuate from paper to paper. Some studies find them distantly related, while other papers find a much closer relationship. In another example, pseudoscorpions, Acariformes, and Parasitiformes are apparently pretty hard to organize because they experience a higher rate of background mutation and evolutionary change than other arachnids. Earlier studies didn't take this into account, and their proposed phylogenies were eventually overturned by later studies that deliberately focused on the slower evolving regions of their genomes. By focusing on these slower evolving, more conserved regions, it allowed for a closer comparison to be made with other arachnid groups, and it produced more accurate phylogenetic models. Okay, now that was a pretty big clarification, but these kinds of background notes and side details are really important. Alright, I think that was a pretty decent introduction to these two groups, so now let's turn our attention to just one of the groups at a time and really dive deep and take a really close look at them. Let's start with the Acariformes, the larger and the older of the two superorders of mites. Within this Acariformes superorder, there's two major divisions, the Trombitiformes and the Sarcoptiformes. Unfortunately, if you dig any deeper into Acariform taxonomy than this most simple basal sister lineage division, you start running into a paucity of data. There just isn't enough consistent data to provide a clear picture of how individual genera and families are organized. For example, there's actually two more divisions of Acariformes, called Endiostigmata and Areophyoidea, but they're both frustratingly poorly defined. They're both suspected of being paraphyletic clades, meaning these are groups that may include actual species or groups of species that are not directly related to each other. Endiostigmata, for example, appears to be a waste bin taxon, where taxonomists took the early primitive Acariform lineages and just dumped them all into Endiostigmata and hoped for the best. But it turns out that members of Endiostigmata include relatives of both Trombitiformes and Sarcoptiformes. It's just a really big mess. The question that comes to mind is, what do we know? Enough about changing taxonomies and mysterious evolutionary histories. What do we actually know for certain about these small arachnids? Well, we know that the Acariformes are small-bodied, globular-shaped creatures with 6 to 8 legs that inhabit virtually every known dry land habitat on Earth, and some marine habitats as well. The two major divisions within the Acariformes are the sucking Trombitiformes and the chewing or biting Sarcoptiformes. These adjectives refer to how they ingest their food. The Sarcoptiformes division, for example, are largely microherbivores that eat small particles of plants and fungi, and detrivores that ingest particles of dead tissue and/or dung. So these Sarcoptiformes bite the food source, they bite off a piece, and they chew on it. The sucking Trombitiformes are fluid feeders. They use a proboscis-like mouthpart to suck up various fluids, like nectar of a plant, or the sweat, the oils, and various secretions, and even the blood of animals. About 4/5 of all species of chewing Sarcoptiformes belong to the Orabitida clade, which is definitely worth taking a closer look at. The Orabitida, also known as the moss mites or the beetle mites, are generally found in moist forest soil and leaf litter, where they chew through detritus, rotting leaves, and the bodies of dead animals, small and large alike. In this way, the Orabitida play an ecological role that's roughly analogous to an earthworm. They break down dead biomass accumulating on the forest floor, facilitating the breakdown of complex biomolecules and the nutritional rejuvenation of the soils, which in turn facilitates plant and fungal growth. However, the Orabitida mites aren't restricted to humid forest habitats. The clade is immensely diverse and has adapted to almost every terrestrial habitat there is, from hot, dry, sandy deserts to cold, lichen-carpeted tundra. Some species have adapted to specific plant hosts, even aquatic plants, where the resident mites have adapted to an aquatic lifestyle. While many Orabitida are microherbivores or scavengers, a small number of them are predators that hunt teeny tiny prey like nematodes and tardigrades. As they get preyed upon in turn by larger arthropods, birds, lizards, and the like, some species have even evolved a mineralized exoskeleton hardened with calcium carbonate or calcium oxalate. Curiously, despite their vast diversification into all manner of terrestrial ecological niches, there isn't a single Orabitida species that's a known parasite. But if we step back to look at all of the Sarcoptiformes again, we'll notice a small clade tucked away in the corner called the astigmatina. These guys, in contrast, are almost all parasites, and their preferred hosts are land-dwelling vertebrate animals, including humans. Within this astigmatina clade, one of the more notorious and hated groups is the Sauroptidia, which includes species that burrow into the skin, or embed themselves in feathers, or that eat hair, or an oily secretion, or some other product of the animal host. Here's a quick, interesting fact. Despite mammals emerging before the birds, the Sauroptidia mites first evolved to parasitize the birds, but then they underwent a secondary burst of diversification, or a secondary radiation, which saw many species adapt to parasitize mammals. Why they parasitized birds first and then went to mammals, even though the mammals were around the whole time the birds were, I don't know. That's still a mystery. Another Sauroptidia family are the feather mites of the Analgoidea, which prefer living on the skin and in the feathers of birds, usually around the neck and the head. Their chewing activities can cause skin irritation, the formation of rough, scabby, or scaly patches, and mange. If the skin damage progresses, these mites can destroy feather follicles and lead to the loss of feathers. Sometimes the itching is so bad that the animal will debride itself of feathers in its attempt to scratch the itchy spot. If the mites inhabit the wings and cause itching and follicle damage there, any feather loss could potentially constrain or even eliminate the bird's ability to fly. Other groups, like the Pyroglyphidae dust mites, live in the burrows and the dens of terrestrial animals, including the nests of birds, where they feed off of flakes of skin or scales, bones, and feathers. Proper dust mites that feed on this detritus and don't actually parasitize the living animal itself are evolutionary descendants of actively parasitic mites. At some point, some ancestral parasitic mites adapted to still eat skin flakes, just without needing to live on the animal itself to do it. They adapted to a more scavenging kind of lifestyle, where they could trail behind the animal instead of riding on it and feeding off of the animal parasitically. There's another Sauroptidia clade, a genus known as Coreoptes. This genus includes several species of skin mites that typically inhabit the ears, lips, and eyes or eyelids of herbivorous animals, like cattle, horses, goats, moose, and pandas. They chew on the skin and the hair of the animal, often destroying fur and leaving bald patches that can also cause itchy patches and erythema, or reddening of the skin, as well as the formation of crusty residues, and they can even have negative effects on the animal's sleep and appetite. Skin mites seem to really prefer the environment of the mammalian ear. It's partially enclosed, it's filled with hair and skin folds, it's warm and moist, and there's plenty of oils being secreted, and gunk getting caught in the hair that the mites can use for food. In fact, many of these mites are grouped together under the informal label ear mites because they seem to almost exclusively inhabit the ears of vertebrate mammals, particularly household pets. Dogs, cats, ferrets, and rabbits, among others, are all very vulnerable to ear mites. Apparently, something like 90% of cats that go see the vet for a case of ear mites are diagnosed with an infection of Otodectus cynotus, which is a particularly obnoxious ear mite that causes extreme irritation and inflammation. As the animal scratches its ear or tries to deal with the discomfort somehow, this can tear up the tissue and lead to opportunistic ear infections. This is actually a really big issue with all kinds of skin mites, just skin mites in general. Through their metabolic activity, they can create skin lesions, and these in turn become portals for pathogens to enter the body. Even normal skin bacteria that would normally reside on the outside, it can get in through these breaks in the skin, these lesions, and this can lead to some pretty nasty infections. You also have to understand that as the mites are sitting there eating skin and forming lesions on the animal's body, the mites are living where they're eating, and they're also defecating and shedding their exoskeletons. And all of this detritus, this mite poop and the mite exoskeletal shells, all of this also contaminates the lesion and can lead to bacterial infection. One of the more common infections caused by mites is scabies. The particular culprit is Sarcoptes scabii, a small, ball-shaped mite with short, stubby legs. When a female scab mite is pregnant, she'll find a warm mammal to live on. Using sharp mandibles and bladed surfaces on the front legs, she'll slice into and burrow under the outermost layer of skin, creating S-shaped tunnels in which she'll lay her eggs, usually at a rate of about 3 to 5 a day, and around 30 eggs or so per individual scab mite. The presence in the skin of the mother mite, which usually dies and decays in the skin tunnels, and the eggs themselves, which quickly hatch into larvae, will usually cause an intense allergic reaction that involves redness and extreme itchiness. In extreme cases, like in animals with weakened immune systems, the mites can reproduce like crazy inside the host and produce huge colonies that permeate the skin, which causes crusted scabies, an advanced form of the disease where skin all over the body is thickly inflamed, scaly, and cracked like the mud in a dried-up lake bed. The dermal tissue is mildly itchy all over the body, and it's absolutely riddled with mites. All of these chewing Sarcoptiformes, from the Orobitida to the Sauroptidia, are called that because they chew their food. They bite or cut out small chunks of biomass, which are then directly ingested. In contrast to this, we have the sucking Trombitiformes, which prefer to consume fluids that can be sucked up through some kind of proboscis. The Trombitiformes include about 1.5 times as many species as the Sarcoptiformes, and all this divergence has led to greater physiological and ecological diversity among the Trombitiformes. A little less than half of all known Trombitiform species belong to a suborder called Prostigmata. Now, if you're a farmer or a gardener, there's a pretty good chance that you're familiar with the mites of the suborder Prostigmata, and there's also a pretty good chance that you absolutely hate their guts, because these are some of the most prolific plant parasites on the planet. The damage they cause to agricultural crops is tremendous. The damage they inflict on living plants in the wild is probably immeasurable. These include the ornate peacock mites of the Tuckerellidae family, which parasitize citrus fruits and grasses in tropical habitats, and the slow-moving, red or brown-hued flat mites of the Tenwepalpidae family, which prefer feeding on the nutritious leaf tissue nearer to the plant's veins. These are related to the spider mites of the Tetranychidae family, which also feed on the softer underside of plant leaves by puncturing plant cells and then gobbling up the juicy inner contents. Individual spider mites don't cause much damage, but large colonies can riddle a plant's leaves and utterly destroy its ability to sustain itself with photosynthesis. Spider mites are so named because, much like their more distant spider cousins, they have the anatomy to produce silk and can spin it into rudimentary webs running along the underside of a leaf. This webbing is like an artificial layer of skin over the leaf that protects the spider mites, and strands of silk can be laid down connecting leaves together, so the spider mites have pathways to move around the plant and get from leaf to leaf. I should mention that the spider mites are highly fertile. Females lay hundreds of eggs over their brief 2 to 4 week lifespan, so they can produce large colonies and can rapidly adapt to environmental stressors. Perhaps the most notorious spider mite is the red two-spotted mite Tetranychus urticae, which lives around the world and eats hundreds of different plant species, from greenhouses to tropical jungles to boreal forests. Here's a particularly interesting fact about the red two-spotted spider mite. Alongside several species of aphid, the Tetranychus urticae is the only animal that can synthesize carotenoids. These yellow, orange, and red-colored organic pigments are naturally produced by many bacteria, algae, plants, and fungi. Genetic research has revealed that the Tetronychus urticae has this carotenoid synthesizing ability due to horizontal gene transfer from a fungus, presumably one that was being consumed for food, but left behind genetic residue that wasn't fully destroyed by the spider mite's digestive system, but instead integrated itself into the mite's genome. The prostigmata trombitiforms aren't just plant parasites. They include many animal parasites too. You've probably heard of chiggers or berry bugs, which belong to the genus Trombicula. In their larval stage, these mites parasitize many different vertebrate animals, from mammals and birds to reptiles and even amphibians. They'll melt a hole in the skin, softening it and creating a space where the mite can dig in and readily chew up cells in deeper layers of the dermis, which causes extreme irritation. After they grow past the larval stage, they'll drop off their animal host and spend the rest of their mature life as typical microherbivores with a diet of plant and fungal parts. Other trombitiform animal parasites, and even predators, can be found in the Scaletoidea and Rhaphignathoidea superfamilies. Okay, so we've talked a lot about the Acariformes. They include many parthenogenic species, and they include among their ranks the Sarcoptiformes and the Trombitiformes, which are both mostly microherbivores with a sizable selection of predators and animal parasites. Now let's look at the other major superorder of interest for today's episode, the Parasitiformes. Remember, these are not directly related to or descended from the Acariformes, as there's several other clades squished between them, but they appear to have evolved into a very similar set of ecological niches, and they have roughly similar morphology. The Parasitiformes include over 12,000 known species, which are distributed unequally among 4 orders. From oldest and most basal to youngest and most derived, these are the Opiliocarida, the Mesostigmata, the Holothyrida, and the Ixodyda. As is tradition, let's start with the oldest clade, in this case, the Opiliocarida. This is a cryptic group of mites. They're relatively large compared to the sub-millimeter-sized mites that dominate the Acariformes, and the Opiliocarida are similar in shape and appearance to the Opilionese harvestmen, the daddy longlegs, although the Opiliocarida tend to have longer abdominal segments and relatively shorter legs, and as a lineage, as a distinct clade, the Opiliocarida only emerged in the mid-Cretaceous, which makes them much younger, hundreds of millions of years younger, than the Opilionese. Even though they're relatively young, not a lot is known about them. Despite being relatively large, they're really rare. Very few specimens, living or fossilized, have been found. Those that have been found all carry several primitive features, including 6 pairs of eyes, instead of the 2, 1, or even 0 pairs of eyes that most other mites have. These primitive features are strange, considering the relative evolutionary youth of not just the Opiliocarida, but all Parasitiformes. The next clade, a bit further down this branch of the family tree, is the Mesostigmata. This is, by far, the largest parasitiform clade, including over 2/3 of all parasitiform species. It's not really that surprising, then, that this clade is also very diverse, including many different predators and a variety of parasites adapted to a wide range of animal hosts. They're actually so diverse that one of the only traits that they all have, aside from typical arachnid stuff like 8 legs and chelicera, is a single pair of spiracles for breathing, positioned on either side of their blob-shaped bodies. Almost every other anatomical feature has been shaped or modified by evolution to such a degree that comparisons of these Mesostigmata species find that they're usually not even remotely alike. So the Mesostigmata order is the largest parasitiform clade. And within this order, the largest clade is the Phytoseidae family, which includes over 2,700 described species. There's a neat little classification scheme for this family, which organizes species into one of 4 types based on their diet, and with further type subdivisions based on type of prey, as well as habitat. 2 groups are generalist predators that will eat a variety of small arthropods, including smaller mites, but one of these groups generally prefers and is most successful at eating pollen and other plant biomass. The other 2 groups are not generalists. They're specialized predators that, depending on the species, hunt certain kinds of web-spinning tetranychid spider mites. Another mesostigmata family is the Veroidea, which contains a monstrously harmful species called Varroa destructor, or the Varroa mite. These Varroa mites attach themselves to the legs of a bee and use them as a vehicle to sneak into a hive. Once these mites have infiltrated the beehive, they'll slip into a brood cell and lay their eggs on the larva. The mite eggs and the bee larva will mature at about the same time, and when the bee larva hatches and leaves the brood cell, the newborn mites will also leave to infect new brood cells and new bees. Sometimes the larva is just eaten from the inside out before it has a chance to hatch out of the brood cell. With adult bees, the mites will typically attach themselves on the side or underside of the bee's abdomen. They'll suck out its body fat, disrupting the bee's hormone signaling systems and immune system, among other things. This weakens the bee, making it much more susceptible to disease. Unfortunately for these bees, Varroa mites are prolific disease vectors, known for carrying no less than 5 viruses that can infect bees and destroy entire hive populations. The stress of a Varroa mite infection, as well as potential secondary infections from transmitted viruses, can cause deformations and lifelong problems for the bee, including a shorter overall lifespan. One such virus is called DWV, or deformed wing virus, which disrupts the bee's memory and ability to learn, in addition to the titular deformation of the wings that precludes any ability for the mature adult to fly. The downstream effects of this, of multiple adult bees, drones that are supposed to care for the young or go out and find pollen and nectar to bring back, the downstream effects of this on all of these bees can be catastrophic for bee colonies. Varroa mites are highly dependent on their bee hosts. When bee colonies are available, Varroa mite populations can grow explosively, exponentially even. But when there's no hives available, they struggle and their population numbers crash. This can happen if the mites destroy their host hive with disease, which is an unfortunately frequent possibility. They've certainly earned their name, Varroa destructor. Now in this line of talk, there's a lot more to explore, like the Uropodidae family, which includes many reptile parasitizing tortoise mites, the Diplogyneidae family, which includes many species that parasitize tropical beetles, and the Viagaidae family, whose species are not parasites bound to their host, but are instead free-living predators that hunt around in the soil and leaf litter or in rocky coastal landscapes where they seek out smaller mites and tinier arthropods to gobble up. The third oldest parasitiform clade is the Holothyrida. This is a really small clade with a mere 25 described species, but all of them, much like the Opiliocarida, are rather large compared to the average mite species. They have very stiff, reddish, rust, brown, or black-colored exoskeletons, which helps them blend into the leaf litter and soil as they prowl around, seeking out their arthropod prey. The Holothyrida have a rather disgusting diet. Their arthropod prey typically isn't alive. They prefer their prey to be dead. Recently dead, too, so they haven't dried up yet, because the Holothyrida prefer to suck out their bodily fluids and various juices. Bigger arthropods, like caterpillars or beetles, make for excellent meals. As the Holothyridae are a sister group to the ticks of the Ixodiida order, it's believed that ancestral ticks originally also ate like this, sucking up the fetid juices of decaying arthropods, before adapting to the juices and fluids of living animals, i.e. their hemolymph or their blood. That's a nice segue into the final division of the Parasitiformes. This most recently derived order is the Ixodiida, more commonly known as the ticks. These are relatively large mites, at 3 to 5 millimeters in length, which feed almost exclusively on the blood of larger animals, like mammals, birds, and less frequently, amphibians and reptiles. They require the nutrition and energy from a blood meal in order to molt and grow into a new instar and a new life cycle or life stage. The oldest tick fossils are about 105 million years old, but genetic evidence suggests they have a far older origin, potentially as far back as the earliest days of the Triassic, 250 million years ago. In this time, the Ixodiida have split into 3 major lineages. The most basal and primitive of these lineages, which is estimated to have emerged about 195 million years ago, has been almost entirely wiped out. Every single member species is extinct, with just one exception. The Nataliella namaqua, which lives in southern Africa, in dry mixed tree and shrub savanna, where it parasitizes pretty much any animal that wanders by, be it a bird, a lizard, or a desert fox. The other 2 lineages are the Argacidae, which includes about 200 species of soft ticks, and the Ixodiidae, which includes about 700 species of hard ticks. There's a number of subtle physiological differences between these 2 groups, but in a nutshell, the titular difference between the hard and soft ticks is the material on their outer dorsal surface. The soft ticks have leathery skin that can flex and stretch as they fill up with blood, while the hard ticks have evolved their scutellum, a hard protective armor plate, to cover most of their back. And of course, this makes them a little less flexible, a little more protected, but less flexible, less able to stretch and engorge themselves with a blood meal. One of the most interesting things about the ticks is the variety of host strategies they've evolved as part of their life cycle. Now, the simplest strategy, for most parasites, not just the ticks, is to use a single host. One host ticks will spend most of their lives growing, feeding, molting, and mating on one host animal. They'll only leave the host animal to lay their eggs, and the newborn young will seek out a new host of that same species pretty much immediately after hatching. The one host strategy is pretty straightforward. The 2-host strategy is similar, but a little more complex. As a nymph, the tick will drop off the host and molt into adulthood in seclusion, before seeking out a second host to find more mites to mate with. To put it another way, the 2-host strategy involves one host that acts like a nursery or a hatchery, while the second host is more like a bar or a party. And then they'll drop off of that second host, try and find the first host to lay the eggs, and then the cycle begins all over again. The 3-host strategy is also similar, except the tick will drop off of the first host to molt into a nymph. Then it'll find a new host, the second host, it'll feed, it'll grow larger, and then it'll drop off of that one to molt into an adult. And then it'll find another host, the 3rd host, where it seeks out a mate, before dropping off that host again to find a new one to lay eggs. So here, our 3 hosts are the animal that's used as a nursery to lay the eggs and hatch the larvae. Then you have that new second host that's kind of like a gym or a mess hall, where the mites go to grow and get bigger. And then the 3rd host is the party, or the bar, again, where they go to seek out a mate, and then reproduce, and then they drop off to go lay their eggs again, and the cycle starts again. The whole time, on every host, and for its whole life, the ticks are just slurping up blood. The initial bite can often be painful, but evasin proteins in the tick's saliva can soothe the immune response to prevent inflammation, and this lets the tick feed without being noticed. Because the diet is exclusively animal blood, ticks end up eating a lot of protein and a lot of minerals like iron and sodium, but they get relatively few sugars and fats. This is a problem for the ticks because they need sugars and fats for energy, for metabolic processes, for their cell membranes, for their neural system, for tons of stuff. So how do they get it? How do they get the carbohydrates and the lipid molecules they need? Well, it's super fascinating, but it turns out that they've evolved relationships with endosymbiotic bacteria, which, in turn, produce the chemicals they need inside their bodies, like steroids or vitamin B. These endosymbiotic relationships have been a major driver of tick evolution, as the number and type of symbioses determines what non-dietary nutrients are available to the tick. If it doesn't have the symbionts to produce a certain nutrient that it needs, that's going to provide strong evolutionary pressure for the tick to find a new host, one whose blood may have the nutrients that it needs, or at least some viable chemical precursor that can be transformed into the nutrients it needs. Furthermore, these endosymbiotic bacteria can be passed vertically from mother to offspring tick, but horizontal transfer is also possible. These mechanisms, which allow the symbiont community in a given tick species to change drastically sometimes, it adds an important degree of instability to the nutritional dynamics of tick populations. In many cases, if a gut symbiont were to die off inside their gut and they weren't able to convert blood nutrients into certain vitamins or lipids or whatever, this will stress the ticks, and it will drive them towards divergences, towards adaptations, where many splinter lineages will go extinct, but others will thrive and establish new populations. They might even adapt to new hosts with new biochemical and nutritional environments. And that, I think, is a good place to wrap up this episode on the Acariformes and Parasitiformes. This was a pretty wild episode, covering almost all major aspects of mite existence, from their life cycle and reproduction, to their diet and habitats, to their ecological functions. If I could summarize these animals with just a brief paragraph, I would say that all of these species generally fall into two camps: the microherbivores, which eat pollen, fungi, and all manner of plant parts, and the carnivores, be they animal parasites, active predators, scavengers, or dung eaters. The animal parasites are prolific transmitters of disease. The predators are useful top-down regulators that keep their prey's population numbers in check, and indirectly protects local vegetation. The herbivores both feed on plants and are themselves food for larger animals, and the scavengers and coprophages help break down decaying biomatter. In fact, that's perhaps the most critically important ecological function of the Acariformes and the Parasitiformes. Their breakdown of dead biomass plays a critical role in nutrient cycling, in rejuvenating soils, and making the habitat more chemically fertile for other life forms. Although they may seem inglorious and, well, kind of gross, the Acariformes and Parasitiformes are critical to our world's ecology and the maintenance of healthy biomes. If you enjoyed this episode, hit that like button or give it a 5-star review. Hit that subscribe button and the bell so you can get a notification right when I post new content. In the next episode, we'll explore the scorpions and their relatives, as well as a few other assorted arachnid lineages, so be sure to check it out. If you want to support the show in other ways, check out the official store, go to the Patreon page and become a Patreon supporter. And as always, thank you so much for listening.